Jaci Saunders. All right, it is my pleasure to introduce Jaci Saunders. Jaci is giving a seminar to join IOB as an affiliate, so this is a normal part of the application process. And Jaci is in my department in marine sciences, so I'm really happy to introduce her. So Jaci's work focuses on how the availability of different chemical elements influences the geographic distribution and evolution of microbial communities. And conversely, how marine microorganisms biochemically transform compounds and influence global elemental cycling. Most of her work is focused on the toxin arsenic, as well as nutrients, phosphorus, and nitrogen. And Jaci does a mixture of hypothesis-driven research and software development, including databases. She works with the semantic web. She has open data portals. So she's very computationally knowledgeable. And Jaci obtained her MS and PhD degrees at the University of Washington, and has also done a stint as a NASA postdoctoral fellow, as well as a Gordon Betty Moore semantic data postdoctoral investigator. And now she is an assistant professor in the Department of Marine Sciences. So, hand over to Jaci.
Thank you, Holly, and I apologize for running late. My life is chaos right now. I have a 7-month-old and my husband is in Spain, so I apologize. It was a rough morning. Anyways, thank you, Holly, for that lovely introduction. I'm not going to talk about the semantic web today. I'm going to focus instead mostly on some proteomics work that I have done. So here we go. So pretty much as Holly already said, my general theme of my research focuses on trying to understand how elemental distributions in the ocean influence marine microbial distribution and the evolution of those microbial populations. And then conversely, I want to know how the marine microbial communities influence then global elemental cycles. So today I'm going to focus on some metaproteomics work that I've done and trying to understand what can metaproteomics tell us about the dynamics between these marine microbial communities and global elemental cycles. First, I want to acknowledge my collaborators, Max Saito. He was my postdoc advisor at Woods Hole Oceanographic Institution, as well as Don Moran and Matt McKilvin, who were part of the lab there. Sanjay was a student who worked with me and helped develop some visualization tools that I'll show a quick snippet of at the end of the talk. Danny Kincaid, Adam Shepard, who helped me a lot with something called the Ocean Protein Portal, which I'll talk about. And then Chris DuPont and Drishti Kaul from JCBI, who helped do some of the metagenomic work on that's essential for metaproteomics data. So a brief overview of my talk. I'm going to go over what is metaproteomics. I'm going to talk about the proteomes research expedition. And then finally, I'll summarize with some conclusions, as well as give you a little snippet of my mentoring philosophy when it comes to graduate students. So with metaproteomics, I'm going to try to explain how we identify proteins with mass spectrometry, and then how we can characterize these proteins via taxonomy and function. So this is just a high-level overview of microbial omics in ecosystem studies. We have the central dogma here. We have DNA encodes RNA, which is then the template for proteins. And then we can look at the substrates and investigate metabolites, metabolomics. So DNA gives us an idea of the microbial potential. So what can a microbe do? What does it have the instructions to do? RNA and proteins, when we look at those levels, we're able to get an idea of microbial function. So what are the microbes doing in a community? A big difference between looking at RNA and looking at protein is the time scale that these molecules exist on. So RNA is more ephemeral, right? Very short time scales, short turnover, whereas proteins are longer. They're stable for longer. There's a longer period for turnover there. And so if you want to say, look at how an organism is going to be responding to a straight perturbation right away, you could look at the transcriptomics, right? But if you, for more kind of like stable, long-term effect, you can look at what type of proteins are being produced by that community. And then metabolomics is looking at the output. So what are the chemical intermediates in metabolism that are being produced? So for this talk, I'm going to focus on proteins, but you'll see how we do use genomics in order to identify the proteins as well. So this sample, the samples I'm going to talk about today come from the proteomes research expedition. And so here's an image of some of my former lab mates deploying these large McLean pumps. So these McLean pumps, you put them out into the water, and you see how they have these two weird little heads on them. They're basically pulling water in through, and then filtering out through a series of filters that are stacked in these heads. And so this allows us to sample hundreds of liters of seawater in a pretty hands-off manner. So prior to my postdoc, most of my filtration at sea involved a lot more blood, sweat, and tears, crying over small peristaltic pumps, if anybody's used those, and just being like, filter, please filter faster, I want to go to bed. The McLean pumps, great. So what we do is we attach them to a line, and we lower that line off the side of the ship. So in this case, for this particular study, we had 8 different pumps. And so you could connect the pumps at different depths along that line, and then you let them hang out for 4 hours, and they just pump. They filter for 4 hours, and you can program the pumps to turn off at particular times or once a certain volume of seawater has passed through. And that's 4 glorious hours where you can get some sleep. So once we take the pumps back onto deck, we can take the filters off. And so this beautiful green color, this is the phytoplankton. So those photosynthetic organisms that we're collecting on this filter. So this is coming from the deep chlorophyll max, which is this like super special point in the water column where you have enough light, most amount of nutrients they can have, and the phytoplankton, this is where you're going to find the maximum density of the phytoplankton in the water column. So if we take all of the different filters and we stack them, we take little punches of them, you can see the color change through the water column. So here's the very surface. You can see it's still green, but it's a lighter green. And this is that deep chlorophyll max sample. So it's a little bit below. It's not exactly at the surface. You move down, and then we go to our very deep sample. So this sample was actually taken at about 800 meters depth. And you can see that there's this big color change because there's less biomass and there's less of these photosynthetic pigments being captured on the filters. All right, so how do we go from a filter to a beautiful metaproteomics data set? Well, we take the filter, we extract off the proteins, extract all the biomass off of that filter, and then extract out the proteins from that. And we're able to measure then how much protein we collected on that filter. This is going to be important for later. So we have all of this protein that we've collected off the filter. We know how much protein we have. We then take that protein and we digest it with trypsin. So we do this tryptic digest, and that trypsin is going to cleave at specific points along that protein sequence. We then inject 5 micrograms of peptides onto the HPLC, and we used this tandem HPLC technique for separation over an 8-hour period. And then we pass it into our Orbitrap. So here is just, a very small peptide chain. And this is the spectra that we get out of our mass spec. So how do we convert this into identified proteins where we can say, what are the taxa? What functions are those proteins? Well, we know that this trypsin is going to digest these peptides at specific residues. So we have this tryptic digest, and we've now digested this longer protein into smaller individual peptides. So an entire full-length protein won't fly through a mass spec. It's too large. And so you have to digest it into these smaller parts. So we have these smaller individual peptides. The way that we then identify from this spectra here to what sequence of peptides we're looking at is to actually reference a metagenome. So in this particular type of mass spectrometry, we're using data-dependent acquisition, which means that we are referencing a metagenome. So we're referencing some template of sequences. So we have our metagenome, which generated this predicted spectra. So we have metagenomes of all of the open reading frames from this metagenome taken from the same location. And then from that metagenome, we use tools to then generate a predicted spectra given the amino acids, theorized tryptic peptides that are digested from that metagenome to generate predicted spectra. And then we match our actual measured spectra from the mass spectrometer against this predicted spectrum. So when we have these matches, we now say, we know what this amino acid sequence is, and we have what we call a PSM, or a peptide spectral match. And so if you look at proteomics literature and they talk about spectral counts, this is what they're doing. Every time you have the spectrum that's matching against a predicted spectra, that's going to be a PSM and give you a spectral count. So one of the challenges with proteomics is that you're only identifying these smaller, shorter amino acids. There can be some issues with this, right? So in this case, we have this one peptide coming from a larger protein. And we know its sequence, okay? We have that peptide spectral match. However, this sequence could be coming from an alpha proteobacteria, but we also have the same sequence, that same short amino acid being found in gamma proteobacteria, right? So we have this challenge of shared peptides. So how do you know what organism this peptide came from? Or even what functional protein this peptide came from? For the most part, this issue of shared peptides is more of a problem when trying to resolve taxonomic origin than functional origin. There is still some issue with determining the function of the protein, and there are some shared peptides between different functions. But we see it a lot with trying to identify taxonomy. So what we did was generated this software called MetaTrip. And in order to calculate out the least common ancestor. So for this given peptide, we know that we see this in alpha proteobacteria, we see it in gamma proteobacteria. So the least common ancestor for this peptide is going to be the next taxonomic rank that's higher than that. So it'd be proteobacteria. So when we're evaluating our proteomics data, we have this peptide found in the alpha proteobacteria and the gamma proteobacteria. The lowest resolution that we can call this sequence at is going to be then proteobacteria. And we do this by passing it through this MetaTrip software that we created. So the MetaTrip software calculates out the least common ancestor of peptides. So this link here takes you to a web portal of MetaTrip. So if you wanted, we have it set up to do mostly marine microorganisms. So we have some metagenomes that have been assembled. We also have kind of standard fully sequenced genomes, and we have MAGs as well. So we're kind of running the whole list between metagenomes, MAGs, and what fraction of peptides show no matches to existing bacterial proteomes? Great question. Anywhere, usually about 25%. Some depending on the metaproteome, the environment you're looking at, but 25% on average, we aren't able to assign. So, so anyways, yeah, we have our metagenomes, our mags, and our, normal full sequenced genome. And we do this for a couple different reasons. So we know the taxonomy when we have an organism in culture and we've been able to sequence the full genome of that organism, we know exactly what the taxonomic attribution is going to be. When we're making these calls in a metagenome that's assembled from the environment, the taxonomic identification is going to be a little less certain, right? And with a MAG assembly, so a metagenome assembled genome, it's going to be somewhere in the middle with your certainty of assignment of that taxonomy. So we've provided basically these 3 separate pools that you can search on in MetaTrip. So that is here, you can search the marine. Also connected to this, we have a coronavirus version. So during the pandemic, my postdoc and advisor and I were, just hiding out in our homes, just frantic as to everything that was going on around us and frustrated with family members who were hearing other things about like, it's just the flu. And we're like, it's not the flu. What can we do? And so we took MetaTrip and we made it appropriate for coronaviruses. And so we analyzed MetaTrip for coronaviruses as well and showed through a peptide analysis, a protein analysis that COVID is not the flu, and it looks very different. Okay, so these challenges in metaproteomics are felt across the board by people working in the marine environment. This is a more nascent field, right? Especially compared to metagenomics, metatranscriptomics in the environment. So back in 2017, my postdoc advisor, Max Saito, put together this working group of people who work on metaproteomics in the environment. So we had people who worked on metaproteomics in the marine community, but we also had people who work in gut microbiome, saliva microbiome, cave microbiome, and we all came together and tried to assess what are some of these challenges that we see when working in these environmental settings with metaproteomics, and what are some best practices that we can come up with. So if you follow that QR code, you can see what the group came up with as our best practices, and we developed best practices, and we also did a large collaborative intercalibration study. So what is metaproteomics? Metaproteomics is the study of proteins produced by complex communities. How are the proteins identified by mass spec? In our data-dependent acquisition, we identify peptides by measuring the mass spec, getting an M over ZN intensity off the mass spec, so generating those spectra, and then matching them against predicted spectra from open reading frames from a metagenome. How do we characterize by taxonomy and function? This is what I'm going to get to next, but we are taking the peptides we've identified, and we're actually pulling the annotations from the metagenome that we assembled. So we're annotating the proteins, the predicted morphs in the metagenome, and then we can transfer that function and the taxonomy once we also keep in mind that lowest common ancestor, that least common ancestor analysis for taxonomic assignment. So now I'm going to talk about the proteomes research expedition. So how do we define biogeochemical regions across the transect? What does the microbial community look at like in this region of the world? What are some major microbial functions with biogeochemical implications that we can identify from looking at this metaproteomic data? Were there any community interactions that we identified? And then I'll get into the conclusion. So this is the proteomes research expedition. It crossed 5,000 kilometers. So that's longer than the distance of LA to New York City. So it's a huge swath of area that was covered. We had 11 stations and we moved, this is station 4. And the way we look at this map, just to orient you, here's the Baja of California on the coast of Mexico. You can see it here. And then we have Hawaii is over here. This is the equator at station 12. So station 4, we transect up, go down across the equator and down to station 14 in the South Pacific. So it ranged from 20 to 1,250 meters deep. We had 98 samples in total. I'm going to present on the 0.2 to 3 micron size fraction. So that is going to capture free living microbes in the water. So things that are 3 micron in size and below. And we again used this untargeted data-dependent acquisition metaproteomics method. And I want to point out that we crossed multiple different biogeochemical provinces, and I'll try to explain a little bit more what these provinces are. So this is looking a section profile that is looking at, we have oxygen concentrations going across this transect. So this is station 4, station 2, or 12 at the equator, going to station 14. And we have depth going from 0 meters and then down to that 1,250 meters. So when we're going across this transect, we can see that in the very surface, fully oxygenated water, right? Fully oxygenated water in that surface ocean. But as we move through depth, we can see that oxygen is being removed. And that's being removed from respiration, right? I want to point out these 2 regions here. We have the ETNP ODZ. This is the Eastern Tropical North Pacific Oxygen Deficient Zone. So this is a region that's naturally occurring, and it's a body of anoxic water that is created from higher rates of bacterial respiration and low rates of water ventilation. So water moving in these mid-layer depths is very slow, and so it's not able to mix and bring oxygen into the water. We have a similar thing down here, Eastern Tropical South Pacific Oxygen Deficient Zone, also naturally occurring. When we look at some of our samples, we have a decent amount of samples that are here captured in that Eastern Tropical North Pacific Oxygen Deficient Zone. So how do we define biogeochemical provinces across this region? You know, it's 5,000 kilometers, a huge swath. So what I did was I took the environmental parameters that were captured along this transect. So the environmental parameters are temperature of the seawater, oxygen concentration, nitrate concentration, ammonium concentration, phosphate concentration. And we took all of these together and we compared them and generated a k-means clustering of these environmental features in order to try to break up this transect into cohesive blocks, into cohesive units. So for the k-means, we first, we have our clusters and we have a series of centroids, and we move the centroids around until we have centroids positioned in a way that most coherently describes this data in a reduced dimensional form, right? We have multiple different dimensions with that environmental data. This is just a way that we can reduce it down to try to cluster it up into different groups. So we, if you look at this elbow diagram, this is non-scaled within cluster sum of squared. And this is just a way to try to visually look at what's kind of an optimal number of clusters, because with k-means, you have to assign how many clusters, how many centroids you're going to use to describe the separation in your data. So if we use 2, you can see at a k-means at a centroid of 2, we have our most dramatic basically descriptor of the separation between this data. So if we look at k-means of 2 across this transect, we have these 2 clusters that come up. And as an oceanographer, this makes complete sense. So this first cluster is associated with the euphotic zone. The euphotic zone is the sunlit portion of the water column. It's that surface ocean. And then the aphotic zone is everything without light. It's below that sunlit area. So it makes complete sense. Sunlight is driving that photosynthetic activity. It's driving that production in the surface ocean. And then at depth, the ocean looks a little bit more similar. Once you get past this euphotic zone and you get into the deeper ocean, it's far more homogeneous than the surface ocean. So we do have this big split between the sunlit region and the dark region. As oceanographers, we know that already. It's not that exciting. There's plenty of work that has been done looking at the differences between the euphotic zone and the aphotic zone. So our next big kind of settled explanatory separation was a k-means of 4. So when we look at a k-means of 4, we have this euphotic zone separation, but we now get differentiation within the aphotic zone. So using this, we were able to coherently break up this transect. So again, this is going to be what we call a section profile, where we're looking across the distance that was traveled on this cruise, going from the surface ocean down to 1,250 meters is our deepest sample, station 4 to 14. So here we've broken it up into these 4 different groups. We have the surface ocean, Klein, twilight, and deep depth groups. And you'll notice in the surface ocean, it's far more shallower in this northern part of the transect compared to the southern part of the transect. If we didn't do this environmental analysis, we would never have known that there was going to be this big shift there. And we identified this big shift, and then it corresponded with a very strong El Nino that happened to be going on at this time. And that El Nino caused the deepening of this surface water of the ocean in this region. So that was pretty satisfying to see the data actually show us this kind of coherent environmental signal. So in order to break up the stations now into some coherent groups, we did hierarchical clustering, and the hierarchical clustering separated out into these very cohesive, coherent clusters, separated out into the northern part of the transect and a southern part of the transect. So we have, I'll be referring to these stations as the north, and then stations 11 through 14 I'll refer to as the south. And I just want to say that because station 12 is the equator, but station 11, which is technically still the North Pacific, actually looks more like the stations that we sampled in the South Pacific. So we captured multiple different distinct biogeochemical provinces across this transect. And the clustering actually lined up very nicely with these different biogeochemical provinces. So the first province we have identified is the mesopelagic. This mesopelagic is this region here, around 250 meters deep and deeper in our particular transect. We also identified the North Pacific subtropical gyre, the NPSG, and that is going to be found right here. So the NPSG is a region off Hawaii in the North Pacific subtropical gyre. So there are these gyres that get set up in our ocean, in the ocean basins, and this particular region is known for very low nutrient concentrations. So the organisms that are living here are working with the minimal amount of resources available to them, and they are scrappy little devils. They have come up with some really interesting ways in order to try to obtain those scarce nutrients, and we see that play out in our metaproteomics data. So we have this region of equatorial upwelling as well. So basically this whole south end of the transect is characterized by having a higher amount of nutrients associated with it. So at the equator, because of Coriolis forces, which are going to deflect things, to the right in the northern hemisphere and to the left in the southern hemisphere, you have this setup where water moves away from each other at the equator, and you have deep water that moves up to fill that surface water that moves away. So you have nutrients that come up from depth in our south stations from that equatorial upwelling. We also have this eastern tropical North Pacific oxygen deficient zone from the oxygen profile I showed you. So that is going to be categorized in this group here. This cluster of samples are more impacted by this oxygen deficient zone here. You can see I've outlined in black kind of the area and the samples that are associated with extremely low oxygen concentrations. You can see there's that little black dot from the Eastern Tropical South Pacific oxygen deficient zone. Unfortunately, we only have one sample in there. And as you all should know, being in bioinformatics, I can't do much with an N of 1. So we're not going to focus on the oxygen deficient zone in the South Pacific. So I'm going to talk about vertical stratification. And when I talk about vertical stratification, I mean, how do these microbial communities change with depth? So when we look at just pure biomass, so now I'm looking at particulate organic carbon that was assessed along this transect, we can see the particulate organic carbon and the vertical stratification of that particulate organic carbon. So at the very surface ocean, you can see that we have the highest abundance of particulate organic carbon. And you'll notice down here in the South Pacific, near that equatorial upwelling region that we have more resources, more nutrients coming into, we see the highest density of biomass. And then as we move down through the water column, we have less biomass. So because this biomass, while there is biomass sinking out of the water column, the majority of it is going to be taken up and utilized in the surface ocean. Only a fraction actually travels down through the mesopelagic, and even less makes it to the seafloor. So now we're looking at total protein concentrations, and you'll notice it lines up really nicely with our total biomass. So this is measured in two different ways. So the particulate organic carbon is just a measure of the organic carbon from the filters, whereas this is a measure of the actual protein coming off those filters. So we had struggled for a long time in metaproteomics, especially in the marine environment, with trying to compare samples in a data set like this that would take into account these massive changes in biomass that occur as you move through the water column, right? So if I'm going to compare this 1,000 meter station, obviously it has lower biomass compared to the surface. And we can see it actually, there's some technical biases that come with this as well. We have an easier time identifying spectra in these deep samples than we do in the way more complex samples in the surface ocean. So in order to be able to statistically compare the proteins that we see in samples from 1,000 meters to the samples of the surface, I calculated out the spectral counts per liter of seawater unit. So I took the amount of spectral counts that we got from any individual sample. So the spectral counts coming off that mass spec after our peptide spectral matches were called. And then compared that with our amount of protein that we injected into the mass spec to get that peptide spectral match. And then I multiplied it by a scaling factor, and the scaling factor takes into account this inherent bias that we see from this technical bias with how many spectra we're able to identify because of the complexity of the samples on the surface versus those at depth. And then we multiply that by the microgram of protein total coming off the filter, because again, we know how much protein we collected on that filter. And then control that for the liters of seawater. So how many liters of seawater passed through the filter to get this amount of protein. And we generate out the spectral counts per liter of seawater. So as I present the proteomics data, the units are all going to be in this spectral counts per liter of seawater unit. So now when we look at our proteins in spectral counts per liter of seawater, we can see that this distribution of all the proteins mirrors that of the total protein we measured coming off the filters, which also mirrors, not exactly, but for the most part, the particulate organic carbon. So how do we define the biogeochemical regions? We used classification models to do it, using environmental data that was collected at the same time that the protein samples were collected. So what does a microbial look like along this transect? So for the most part, the microbial community is dominated by just a few key players that have the most abundant protein signature within this free living microbial community. So here we're just looking at an idealized water column. We have the surface ocean, and this surface ocean is going to be oxygen rich. We have light coming in, so we have photosynthesis occurring here, but because we have all this active photosynthesis, they're pulling up the nutrients and it becomes nutrient poor. At depth, we don't have this sun anymore, and so this particular part of this water column is going to be along this transect, especially, is going to be oxygen poor. We're going to have more nutrients down here. So for photosynthesis, the primary photosynthetic organism along this transect is going to be the marine Picocyanobacterium prochlorococcus. So here is a little headshot with some false coloring. So that green color on the filters that you saw is coming from the prochlorococcus cells that are captured on that filter. So in the surface ocean, and especially just below that euphotic zone, so in that transition area, so this is a region where there's a lot of the biomass, like more biomass makes it here, and then you have biomass being lost as you move through water, as the heterotrophic microbes degrade it, right? So we have Candidatus, we have Pelagibacter up here, also known as SAR11. And they are going to be the heterotrophs here, and they can also use a lot of C1 compounds. Down here, usually kind of around the oxycline, or where we have this big transition in oxygen concentrations, we see Thalmarchiota. What is it now? Nitrosphaeriota, I think, is the new name. The nomenclature, the names keep changing. I think this organism has had about 3 different names in the past 10 years, but the Thalmarchiota are ammonia oxidizing archaea. And so they are going to be chemoautotrophs that oxidize ammonia. And then we also have this nitrifying bacteria called Nitrospina, and they oxidize nitrite. So these 2 organisms are incredibly important to nitrogen cycling in this open ocean water column. All right, so how we characterize the peptides and proteomes. As I mentioned, we compared it to a custom metagenome assembly. So Chris DuPont and Drishti Kaul were the ones who did the sequencing and assembly of the metagenome. So we had over 28 million unique open reading frames in this metagenome. And then we used this metagenome and searched against it, where we identified 107,000 plus unique peptides, which then mapped to 88,000 unique protein sequences. So to generate our annotations, we annotated the metagenome, or the open reading frames. For taxonomy, we used just NCBI-NR, and for function, for this particular analysis, we focused on the KEGG orthology. So using the KEGG HMMs in order to assign functional attributes to the proteins that we identified. This histogram here is showing the distribution of the length of peptides across this data set. So our mean length of peptides was 16 amino acids. We kept the minimum at 8 amino acids, and the maximum size was 54 amino acids. I'm really impressed that a 54 amino acid peptide even flew through the mass spec. So let's look at some data. So again, we broke up this transect into different depth groups. We have our surface water column, our Klein, our twilight, and our deep depth groups. And this is scaled for the amount of biomass found in these different depth groups. So in the surface ocean, 85% of the proteins through this entire water column were from bacteria. And here we're just looking at the taxonomic breakdown for these different depth groups. We have surface, Klein, twilight, and deep depth group. This is the photosynthetic bacterial abundance in the surface ocean. You'll notice it's characterized predominantly, we have this huge contribution from the cyanobacteria. And when we look into the Klein, so the next depth group down, we can see that that contribution of cyanobacteria dramatically shrinks, and we have other groups increase. So we have proteobacterial abundance increase. We now see more abundance of nitrospina, which are those nitrite oxidizing bacteria. And we have thermoproteota, and those are going to be those ammonia oxidizers, also increase in abundance. So when we look at the function of the proteins identified across this transect, we found that out of all of the KO groups that were identified, the abundance of each KO group, over 50% of protein abundance was found with only 25 KO groups. And when we look at what are kind of the overall functions of these different proteins, like are there any characteristic functions dominating any of these depth groups? When we look at the surface, Klein, twilight, and deep, we can see that the surface has a very high relative abundance of transferases, in particular DNA-directed RNA polymerase. So what does that mean? It means that the cells in the surface ocean, they are growing, and they are growing fast. So there's far more energy is being directed and resources being directed towards growth in the surface ocean. And when we look through then the deeper water column, Klein, twilight, and deep, we can see oxidoreductases are our dominant form. And these oxidoreductases are really important for obtaining energy. So if you want, you can explore our data if you care. Each one of these boxes are scaled to the relative abundance of the particular type of, that particular KO group within the surface, Klein, twilight, and deep. And so you can interactively explore the data this way. So we found mostly bacterial proteins in the proteomes transect. 85% of the proteins were bacterial, cyanobacteria dominating the surface. We see ammonia oxidizing archaea and nitrophysers more abundant coming into that Klein community. And we see this focus on obtaining energy at depth and growth in the surface ocean. So again, these are just looking at the transect and our major biogeochemical provinces. So we look at this nitrite oxidoreductase, part of that nitrospina, that nitrite oxidizer or nitrifier that is oxidizing nitrite to nitrate. Previously, we had published data looking at just the northern part of the transect, and we found that they are one of the most abundant proteins in the ocean. And however, though, this North Pacific, once we caught a chance to analyze the South Pacific, we realized that we didn't fully capture the whole picture of distribution of these nitrospina. And so when we looked at the South Pacific, we actually found that they're quite abundant in the surface ocean in the right environmental conditions. Now, if we look at the ammonia monooxygenase, so of the ammonia oxidizing Thalmarchiota, we have ammonia, it's reacted with oxygen, and we find a high level, a little bit deeper down in the water, but still this is mostly considered in the surface, but you can see statistically, there is a strong association of this Thalmarchia ammonia oxidizing protein associated with that region of equatorial upwelling in the South Pacific. So this was an interesting story looking at urease. So the urease protein cleaves off ammonia from urea. So in cyanobacteria, this urease is being used to obtain nitrogen as a nutrient, right? They're using this for nutrition. And we can see that there's this high abundance of urease in this region associated with the North Pacific subtropical gyre. That's a surface region where we have very low nutrient available. So there's not a lot of nitrogen available for these organisms. And so they are doing the work to actually cleave that ammonia off urea in order to use it to sustain themselves. In the South Pacific, if we look at archaea, they are going to be using urease, but this case, they are cleaving that ammonia off for an energetic substrate. They're taking then that ammonia and oxidizing it for energy. So we see that NXR, that nitrite oxidoreductase, is dominant across the transect. We also see that urease has these different distributions based upon the taxa and what the needs of those different taxonomic groups are. So I had all this data, and I needed to understand a bigger picture. What are some of the community interactions? So I used a self-organizing map, and I'm just going to kind of fly through this because I believe we're short on time. And so broke this up into a 30 by 30 grid that was assembled. Each one of these dots represents a protein. So a function from a particular taxonomic group. And where you see it lit up, it means in a given sample, the proteins that are lit up are kind of explaining that sample. They're the most important things driving that sample. So if we look across our transect, we have all of these different maps that were then feature maps we're able to generate for each sample. And they have these unique fingerprints of like what the microbial community is doing at each one of these different samples. So from this, we were able to identify some associations. So if we look at a composite of all of these different features together, we have this, and it shows us alpha proteobacteria, cyanobacteria. And I want to highlight here, we have that ammonia oxidase enzyme, the A, B, and C subunits in these white stars. And you can see all these yellow stars as well that are nearby, and then 2 more yellow stars here. These yellow stars are ammonifying hydrolases produced by heterotrophic members of the community. And so when we look at the ammonia monooxygenase enzyme compared with these ammonifying hydrolases, we have a significant relationship where there is a significant positive relationship between the presence of ammonia hydrolases and our archaeota, or ammonia oxidizers. So their ammonia oxidizers are reliant upon their community members, this syntrophic dynamic, to release ammonia that they can then use themselves for energetic gains. Okay. So I just want to plug the Ocean Protein Portal. This is a web portal that I am a co-I on. And you can check it out here. If you want to explore any of the proteins in this data set, you can go and check it out here. We also have other metaproteomic data sets from the marine environment that you can explore. And Sanjay helped design some of these visualization tools. And so in conclusion, metaproteomics can give a lot of insight into how marine microbial communities are interacting with biogeochemical cyclings and with the interactions within the community. And we identified structural profiling. We also saw horizontal structure when we looked across from the North Pacific to the South Pacific. And we were able to identify some syntrophic dynamics between heterotrophs and archaeal ammonia oxidizers. Finally, my mentoring philosophy. I feel like I'm a cruise director for my students. For the most part, I'm here to help them, provide them with a suite of options and let them choose their path. Everyone is a unique individual with unique goals. And I also think during graduate school, you're changing so much, you're learning so many new things. And so I try to help identify appropriate classes. And so that's one thing that I'm really excited about IOB and getting associated with IOB for, is connecting them with the classes and also the seminar, this community, to help them pursue their goals of using bioinformatics tools. I also support attendance at targeted workshops. I went to workshops when I was in grad school, and have been trying to identify and help support my students to go to workshops themselves outside of UGA, a plug for the microbial diversity course at MBL that one of a grad student that I co-mentor with, Marianne Moran, just got back from, and I swear she was giddy and like skipping when she came back from that transformative experience from what I understand. When I train students in bioinformatics and coding, I encourage them to look under the hood, know how the sausage is made. There's so many R programs and Python programs, especially R. If you put garbage in, you're gonna get garbage out, but how do you know that it's garbage unless you know what's happening in between? What is that program really doing? And so I really encourage my students, and I harass them when they come with a different program, and I say, what is it doing? How exactly is it working? And I also expect them to share their code with me. And occasionally we try to I'll help them debug some code when necessary, but for the most part, I pester them with questions and make sure that they know what's happening under the hood. Finally, I believe in providing a structure of like scaffolded support initially, but I expect them to be their own independent researchers by the end. And so for chapter 1, I believe that they should work on like a really structured problem when possible. And then by chapter 3, I expect them to basically design chapter 3 within, you know, the realm of what funding we have available, but for them to develop chapter 3 by the time they get there on their own and really make it their own and carve their own niche. And then for networking, I'm a cruise director, and I just want to connect them with the right people for them to pursue their goals. And so I currently have 2 students with completely different interests long-term. And so I encourage an internship outside of an R1 academic setting. So one of my students is really into education, so I'm helping facilitate that trajectory. And then, and I just think that R1 research grad students should get a taste of life outside the R1 research sphere. But this might be biased because I had time outside of R1 myself before grad school. All right, I'm sorry for being late. I want to acknowledge all these amazing people, especially the CytoLab folks at Woods Hole who worked on MetaTrip and the Ocean Protein Portal, and my wonderful collaborators. And if you want, there's a video out there of all of the proteins from proteomes. Sorry.
All right, we do have time for a couple questions. We'll start in the room and then I'll check on Zoom. No.
Out of the predicted like ORFs that you had from the metagenome, and then you map the proteins to them, like how much of the metagenome predicted ORFs are covered within the proteome that you got?
Just a small sliver, really. So let me, I've got the numbers somewhere.
Sorry. Yeah.
Sorry. Oh wait, no. Oh, they're separate. Okay. Duh. I have a slide on that. Here we go. So you can see that we had over 28 million open reading frames, but we only had 88,000 proteins that were identified. So a small proportion of proteins are actually identified. You know, given the metagenome though, again, we're looking at 2 different kind of viewpoints of the microbial community here. With the metagenome, we're looking at the genomic potential, and we know the whole genomic potential is going to be expressed at one time. And the proteins are going to be what's being expressed then and there. And there's also going to be just a limit of detection issues as well. It's harder to, you need a higher amount of biomass to get proteins than you do for nucleotides. Yeah.
Hey, that was a really nice talk. I have 2 quick questions. First, either through the mass spec or through the measurement sampling and the prediction of peptides or protein, how often do you find proteins that have unique folds or ones that are not previously in?
Like basically no idea no relation to anything at all. So it kind of ranges. It's about, like, depending on the sample, you can get up to like 25% proteins that we're not sure what they are, where they came from. About 5% of this data set was proteins that we didn't know what the taxa were. We didn't know what the function was at all. And that's really cool to us. We want to explore that. So we see the dark DNA, and then we also see the dark protein. And so that is a component here. That is an exciting component to explore. So we know, though, in these marine environments, especially at depth, that there is that there's absolutely, or that these are unique communities that we just don't have a good reference for. And so I'm really excited about things like the dark protein, because it kind of supports dark DNA and it winnows down that dark DNA into the dark protein. But yeah, it can be up to 25%, especially in those deep samples.
And then, sorry.
I have one more question.
Yeah.
So on one of your slides, you talked about where all of your DNA is coming from, like bacteria, viruses, archaea. You had one section that was from Luca. So what
yeah.
does that mean by that?
So hold on, where did I put that? But no, I went too far. So Luca it's not Luca. It's that when we do our least common ancestor analysis, it means that this peptide is going to be shared across all of these groups. So it's going to be shared across, or basically shared across at least two of these groups. So it would be like archaea and bacteria. So some protein that's shared across both those domains, or shared across viruses. So that's that Luca fraction. And you can see there's also we have this unknown fraction of things that are completely unknown. Usually we cannot, so I guess that's part of how you slice it too, with the 20% identification comes down to also, like, where do you call it? Archaea, do you call just like a bacteria a taxonomic ID? I don't know. Depending on how you slice things. Sure, I can call things as just bacteria, but I don't find that very helpful.
All right, and then one last question on Zoom, and then we're gonna finish up. So in your data analysis pipeline, what happens to the peptides with modified amino acids?
Great question. So when we do our initial searching, we use the program called Sequest. And we are looking at kind of a few common modifications. So we do have a few modifications in there, but there are definitely, you could explore more. We are just using kind of a basic small suite of common modifications, but I know one of my lab mates was really into phosphorylation of amino acids, but that blows up your search time. So we kept it pretty modest.
All right, so I think we're gonna wrap up because there's class after this. So thank you again. Enough for lunch. One straight to the normal room. You don't have plenty of time.
Yeah, I know.